All right. We're so excited. Next, we have our gene therapy and gene editing session. I know everybody is very interested, so we have some representatives from companies in the space today. And we are actually going to welcome those speakers up. You can come on up. And I will have Dr. Bonneman again. He's going to give us a brief primer. So for those families that might be either newly diagnosed or a little new to gene therapy, don't worry, we're going to give you the basics. And at the end of the session, it'll be open for Q&A. So please do jot down your questions as you go so you don't lose them in your head. And Dr. Bonneman, I'll let you take it away. Thank you very much.
Okay, so what I do is I give you a little bit of a high-level overview into gene therapy. I don't have my own slides. These are slides from Cure Duchenne, so I'm just going to use them to guide ourselves along this process. But I try to give you some kind of general principles maybe as an introduction here. So what is gene therapy? So most of what we're going to talk about today is gene replacement, where we put in a replacement construct that is making up for the gene that's defective, in this case Duchenne muscular dystrophy gene, the dystrophin. But there's also other technologies that we can use in gene therapy. There's transcript-directed therapy, so it says gene modification here, but it really is transcript-directed therapies, which is when you go to the messenger RNA. That's the basis of exon skipping therapies, for instance. And then there is therapies that go directly to the genome, to the DNA where the genes are encoded. Those are gene editing, base editing, prime editing technologies that really go to the genome itself. And then we also can do gene knockdown, but that's not relevant for Duchenne muscular dystrophy, really. Gene therapies have a history that is short, but lively, if you will. There are gene therapy products, and this is now AAV-mediated gene therapy, that are already approved by the FDA, that are in clinical use for blindness, a form of blindness, for instance, and then also for spinal muscular atrophy, or GENSMA, in 2019, where AAV vectors have been used for that. So AAV is a magic word. AAV is a workhorse of gene therapy. That's in almost any of the inpatient clinical trials at the moment, where it's done in the patient and not in cells of the patient, so systemic in the patient. That's AAV. And AAV stands for adeno-associated virus. It's called adeno-associated virus because it comes along with another virus in nature. Adenovirus is a big virus. AAV, adeno-associated virus, is a tiny virus. And it comes along with adenovirus. And the reason for that is AAV as a virus has a very small genome. It has a very tiny genome, and it only has 2 genes in it for the virus itself. So it can't rebuild itself from that genome. It needs the help of a big virus. So it kind of co-infects and uses the gene products from the big virus to build itself again and then infect. So it's a small virus that kind of comes along for the ride. It is ubiquitous. It's everywhere in nature, and it's infectious, but it's not pathogenic. And that's a great thing about it. So it infects people, it replicates, but it leaves them alone from an illness point of view. It kind of goes on and does its further infection down with other people. The only downside of that is it leaves immunity. So once you're infected with an AAV, natural AAV, you're immune against that AAV. You can't be infected by it again, which is great in nature, but it's a problem for gene therapy, of course, because if you use it, you're immune to it. There are natural serotypes, meaning natural forms of this virus that infect different tissues and different cell types, including muscle, and we select our AAVs based on the ability to do that effectively. But there are also developments now underway to create in the laboratory new AAVs, new serotypes that are better at going to muscle more effectively and at lower doses. So that's in the works as well. There are certain challenges that come with the size of AAV. I told you AAV is a tiny little guy with a tiny little genome. And the principle of gene therapy is this. You take the envelope of the AAV virus, so the thing that basically has the address written on it, and you take out the genes from the AAV and you put in the gene that you want to deliver. So you basically make in the lab the envelope, the package that you want to use to get to the cells, but you replace the innards, the genes, of the virus with what you want to deliver. And then you have the virus infect once. It can't replicate because it has no genes of its own anymore, but it delivers the goods. But because it's such a tiny guy and its genome is so small, what you can replace is also limited. Only what fits into the natural viral genome in size can be replaced by that. And that's a maximum of about 4,500 bases. For your payload, even less, because you have to put in regulatory elements. But what you want to replace in Duchenne muscular dystrophy is shown up there. That's about 11,500 bases long. So you can't fit that into AAV. It's too big. And even though it's our workhorse, it does not work. So what are the solutions to that? So nature, already clinical observation in nature has told us, yes, this is a really big protein, but you can actually live fairly okay with big part of that protein missing. And that was from observations in patients with Becker muscular dystrophy, a much more mild form of a dystrophinopathy, where in the genome, big chunks of the dystrophin gene were missing, and a much smaller protein was being made, and the disease was still fairly mild. So that led to the concept of developing in the laboratory miniaturized versions of that called microdystrophins or mini-dystrophins or miniaturized dystrophins, where you basically cut out big parts of the inner repeat sequences of the dystrophin gene and make it therefore much smaller, keeping the business ends intact. So the green is the actin binding domain, the orange is the cysteine-rich domain that binds to the proteins in the membrane, and then the red is the C-terminal domain that also turns out to be relatively dispensable. So that was also cut out in terms of saving real estate, if you will. And there was an entire development of these microdystrophins. I think Shinichi Takeda, more than 25 years ago, made the first one, and Zhao Zhao, Jude Samolsky, of course, Jeff Chamberlain, hugely important in that, Longshan Wan, and many others who really systematically over decades developed iterations and iterations of these small molecules, try to find out what are the parts that we can dispense of, which parts are important. And we're left with a number of these microdystrophins that have been now incorporated into clinical trials. You can see they're somewhat different. The business ends are the same, but what's in the middle is somewhat different. There's kind of different arrangements of the flexible regions, the hinge regions. One construct has a NOS binding site in it. And you can see they are different, and they're different by rational design. So every one of these dystrophin constructs has a rational idea behind it, has data behind it, has pros, has compromises in it, and so you can't just look at them and say, oh, this is crap and this is going to work. They all have their pros and cons, and they're all rationally designed to do as good as they can. And this is, apologies to Regenexx Bio, this is an old slide and doesn't even have Regenexx on it. These are some of the constructs that made it all the way, of course, into clinical trials. The Pfizer construct, Sarepta construct, which is the same that's also used by Genethon Solid, and then we have Regenexx also that you're going to hear about. But again, recognize that the principle, the basic principle of all of these is the same. Business ends intact, and the repeats being variably taken out to see that we can maximum get the most benefit that we can get from the small molecule. It's still imperfect. It's not the real thing. The real thing is full-length dystrophin. That would be the ideal thing to have. The other thing that one has to recognize in AAV-mediated gene therapy is if you deliver this miniaturized dystrophin to a cell, it will be in the cell nucleus, but it will not, for the most part, not integrate into the genome itself. So it lives in the cell, it makes it little microdystrophin and does so effectively, very effectively actually. But when the cell divides and has offspring and the nucleus divides, then only one of the daughter nuclei, or less so, the more you divide, the more you dilute out the dystrophin that's been delivered with the AAV, because it lives in little, we call it episomes, it's its own compartment in the nucleus. But when the nucleus divides, it is not replicated. It just dilutes out. And so that's a problem when the muscle grows, of course. If it turns over, then you lose it if it's an episome. If you do editing of the genome itself, if you kind of repair in the genome itself, that edit is inherited, of course, to daughter cells. I mentioned many of the challenges already with the size capacity. the tissue targeting, the doses that you need to get to all muscles of the body, the development in capsids to get better at delivering to muscle, the turnover of muscle that you have to account for when the patient grows, when the muscle turns over, so that the effect is maintained. And then, of course, the adverse events, and maybe I take 30 more seconds here. We are already in the first session talked about that, so I can be very brief here. There are adverse events that have happened that are just related to using AAV as a tool, as a capsid. So certain things happen. You can sometimes get a little of a liver inflammation. Sometimes you can get something like thrombotic microangiopathy, which are all reactions to the AAV capsid. But also what has happened in these trials, what I mentioned this morning, and Dan mentioned this morning, are adverse events that are related to a combination of the transgene and the patient in whom that transgene is given. So that's what we referred to this morning. There were adverse events in a number of boys, five boys in the clinical trials, across the different clinical trials. And what they all had in common when our working group got together is that they had genomic deletion, so they had part of their genome missing that was coding, that was responsible for proteins that are now delivered with the transgene, peptides that are delivered with the transgene. And when you think about what that means is this patient who has a deletion there in the genome cannot make the protein that is encoded by the part of the genome that's now missing. So that patient has never seen this protein in any way, shape, or form. And now we're delivering it with the transgene, so that's completely foreign to the body. It's like you transplant, as I mentioned this morning, transplant an organ into someone who has never seen an organ like this that's foreign to the body. So that patient is immunonaive and is prone to reject the transgene because it is foreign to that patient's immune system. And so that's what happened in these patients. The T cells, the kind of fighting cellular police of the immune system recognized this as foreign and attacked the muscle and the heart where this was expressed. And that has led to the recognition that for now, as we go forward, we have to avoid patients' exposure to the transgene that are immunonaive, that have this immunological risks. And you will see in the trials that are being described coming up that these exclusion criteria are based on an experience like this to make sure that when this is given, it's given safely and no immunological consequences are imminent. Doesn't mean that you cannot address it in a rational way. You can. By understanding it and by developing therapies and protocols and immune modulation that would take into account the individual risk of the combination of the mutation and the transgene to make sure that the transgene is not rejected. So that will come up, that will come, so don't despair by the exclusion criteria. It is just a step in the right direction to make sure that we are moving forward safely and then in the end to be inclusive of everyone. After all, now we can transplant hearts and kidneys in almost everyone by using the right protocols to do this. Okay, so that was the gist of what I wanted to say as a background of the outstanding presentations that will be coming up and will be available for questions after the symposium. Thank you.
Thanks, sir. Gotta give that back. Great. Well, good morning and welcome to sunny San Diego. I imagine you can't tell that it's sunny from inside this room, but I'm really happy that you all invited us here. My name is Teji Singh. I'm from Sarepta Therapeutics, and I was going to cover some of the things that we're doing, focusing on our 9001 program. So Sarepta, our motto obviously is that, we're waiting for the next research, but we can't do any of this without all of yourselves, your families, your patients, and of course the boys and girls that we're trying to treat. This is my forward-looking statement slide. All this means is I'm going to talk about some things here that haven't happened yet. So please don't hold me to everything I say, that they're subject to change. But that's just a disclosure. And of course, my other disclosure is that I work for Sarepta. So what do we do at Sarepta? Well, people forget, Sarepta is actually 43 years old. 1980 is when it was formed, but in the last decade or so, we would focused a lot of our research in the Duchenne space. We have 20 programs overall, 14 in the RNA field with PPMO and PMO with 3 approved products, 4 gene therapies, but I'm going to talk about 9001 today, and 2 gene editing therapies that are in discovery. So one of the things that I also want to point out is that in 2019, in order to actually accelerate our ability to develop 9001 and to get it to more patients globally than we could do it ourselves, we partnered up with Roche, and Roche has the license to commercialize 9001 if and when it gets approved outside of the US. So they'll be our global partners for the commercialization ex-US. Dr. Bonneman's already covered the basics of gene therapy. Now, gene therapy is a bucket. A bunch of things do it. So in Japan, PMO and PPMOs are considered gene therapy. So when we talk about gene therapy, normally we're talking about the traditional things when you give a vector with a transgene in it, but that's actually gene transfer therapy. And I think in that previous session today, you heard it's very similar to not only the transplant field, but gene transfer therapy introduces a new gene into the cells that you're targeting. Gene editing is where you're actually changing the genome within the cells that already exist, either in vivo, so within a person, or in vitro, where you're doing it outside and then giving it back to the people. So you're actually editing it out with CRISPR-Cas9. And then gene silencing, multiple ways. It can be either with one of these gene tools, or it can be with an RNA or an RNAi. So the DNA or the RNA can be silenced. Dr. Bonneman also covered a lot about the vectors. So the vectors that are most commonly studied for gene therapies are AAV, so adeno-associated viruses. They were initially thought to be an artifact. These people were studying adenoviruses and kept seeing this small little dot on the slide, and they thought it was a smudge, or like, how did it get here? And then they figured out this little thing was actually accompanying these big AAVs, to use their machinery to replicate and reproduce. So that's why they're called adeno-associated viruses. They're very, very common to use for a lot of gene therapy programs. There are other few versions, but these have been studied for over 25 years now, have the largest number of exposures across a variety of disease states, lots of different serotypes. And what you do in development is you pick an AAV that you think would be best suited for the disease or the target that you want. So the beauty of these is they're very, very efficient to get into cells. I mean, they're small, but they can get into the target cell depending on the serotype. They're very stable, so when they get in the cell and they unload their package, they're actually incredibly stable to do that. They're not able to reproduce on their own, so they're not directly infectious. They need a partner for that. So if you give it, they're not going to cause an infection. There's no known human diseases associated with AAVs. Rarely incorporated into chromosomes, like Dr. Bonneman mentioned. That doesn't mean they don't, but the ones that have happened in our hemophilia areas are not in any area that's causing any sort of manipulations or mutations within the target cells. The vector that you choose tells you which cell you get, and I'll talk about what our vector is and why we picked it. And there's, pretending again on the vector, there can be low levels of a pre-existing immunogenicity. So some will be a little higher depending on natural exposure, some will be lower, but they vary by vector. So what do we use at Sarepta? Well, we use AAV RH74. The RH stands for rhesus monkey. This is where this vector actually was discovered and isolated from. One of the advantages, or one of the reasons we picked using this is when we were working with it, it actually has really good tropism for skeletal muscle and cardiac muscle. What that means, it actually targets those cells specifically and gets into them. It does get into other cells, but these ones it gets into with a much greater affinity. So that's why we personally chose that. The other thing that's important for here is in our non-clinical package, when we study this, it didn't have any expressed toxicities. Of course, that's non-clinical. That doesn't mean it's the same thing translates into humans. And overall, between our 9001 program for Duchenne, plus our limb-girdles, we've now dosed over 150 subjects or 150 patients with RH74. That's not the whole thing of 9001. So now the big name, which always takes me a long time to pronounce, delandistrogene moxeparvovec. SRP-9001 contains the vector and the stuff inside the vector, so the transgene and the promoter. Now, if you hit that QR code, there's a little slide or a little presentation there that goes over all of this in much more detail, but the vector is one component, and like I mentioned before, this was because of its trophism, targeting skeletal and cardiac muscle. But in it is the transgene, the business end, that leads to the production of the SRP9001 dystrophin protein. So that's the one that actually gives the message to the cell of the protein that we need to get produced. And then in addition to that, there's some other little sequences, but the next most important thing is the promoter. And in our particular construct, we use the MHCK7 promoter. This is a muscle-specific promoter, a little bit more affinity for the heart than skeletal muscle. But what it kind of does is when this package gets in, the vector unloads its transgene, the transgene makes its way to the nucleus. There needs to be something that actually then turns it on, gives the instruction to the cell to start reading this transgene to make this protein. And that's what the promoter does. It promotes that production. That means that you get production in the cells you want to, with lesser production in cells you don't want, or off-target cells, like the liver or the spleen. Dr. Bonneman really nicely pointed out, dystrophin is, the dystrophin gene is the largest gene in the human genome with 79 exons, second largest protein. I know this slide says it's the largest, but Titin's actually bigger. But because it's so large, you can't package it into this small AAV. This little AAV has a capacity of about 5 kilobytes, about 4 kilobases. So dystrophin, the protein, is 3 times larger than that, the gene. So you have to make some choices, and that's what lead to these productions of these smaller versions. And how we came about that is there was this patient, a sentinel patient. There's more than one. Everyone thinks there's just one, but there's several that were identified. But the 61-year-old patient that Dr. Davies identified about 4 decades ago, this patient had muscular dystrophy that was diagnosed well into his 40s, but it was still ambulatory at age 61. He was in the Army, in his 20s, etc. So they thought, okay, what's the basis of this muscular dystrophy? And for sure that he was going to have a small mutation. But when they genomed him and actually looked, he had a large portion of his dystrophin gene actually missing. So that led to this idea that you can actually remove large portions and still retain some level of functionality. So then over a whole variety of experiments with different constructs, not just our company, others, lots of researchers, over 30 different people have been working in this field, they came up with ideas of what were the most important elements to keep. And in our case, basically, we can make some of these choices of what was the most important elements to keep that functionality in there, then brought it down to a size that you could package it into the AAV vector. So that's why it had to be shortened down. So what have we done with 9001? Well, we've actually were able to complete 3 of our clinical studies to put together a package of 85 subjects, 85 patients with safety data and efficacy to file our BLA back in September. So that was last year. And in November, the FDA has accepted that package. So they basically said, we will now give you priority review, means they're looking through the data. In March, they told us, we're probably going to have a discussion with some other external experts. So we had an advisory committee agreement to be done. So the FDA set up. The date of that, not that I'm keeping track, is 499 hours away. So 3 weeks today, on May 12th, we will actually have that committee meeting. That committee will to make it clear, what the committee does is actually give advice to the FDA. The FDA will be the ones asking the questions. And then that advice the FDA will take into consideration on their decision on the product. But the PDUFA date, this is the regulatory date that they have to actually meet based on the submission date, is then May the 29th. So by May the 29th, we hope to hear from the FDA what their thoughts are. See what happens. This package that we submitted was based off our 101, 102, and 103 studies. I think we've covered them in previous forums, but just as a big overview of them, 101 was our first proof of concept study by Dr. Mindell at Nationwide. 102 was our pivotal trial with 41 patients randomized to placebo and control. Hit a primary endpoint of dystrophin expression, the biomarker. Did not hit the endpoint of function. It missed statistically. A variety of reasons for that. Study 103 is our open label study where we actually expanded the population. So we studied boys as young as 3 and as old as 20. We had in that study both ambulatory and non-ambulatory cohorts, as well as a few others to explore some other aspects of the disease. And then study 301, if you get accelerated approval, you have to have a confirmatory study Study 301 is our phase 3 randomized double-blind placebo-controlled study. It's fully enrolled. It'll be the confirmatory study if we get accelerated approval. It would be the basis of a BLA if we don't get accelerated approval. All of these studies are actually enrolled, so they're not recruiting right now. But we do plan in our next steps sometime this, actually in the next 3 months, this summer, June or July, begin our next phase 3 study, our study 303, which will be older ambulatory boys and non-ambulatory boys. So then again, a global study that the protocols will be completed. We've submitted it. We have some local approvals. We're waiting for some approvals with regards to the assays, but that study should be starting at the end of this first half. We're also expanding into a younger population for 2 reasons. One, we're mandated to do a PIP, a pediatric plan for the European side, so we have a study for the younger age group. Because we, as Dr. Bonneman mentioned, we have exclusions based on genetic mutations because of the safety effects. We're trying to expand and learn more about that, so we have an expansion study for mutations. And then we have 2 other studies that are going to be our first proof of concept studies to look at patients with pre-existing antibodies, how we can help to actually remove the pre-existing burden, and if we can successfully give back gene therapy. So if you want to know more, just touch this QR code. Anyone's got more information on all of these things. And what I'd like to do by closing out is thank everybody here. Thank the patients, obviously, everybody that we have in our trials. And I also want to give a shout out to Cure Duchenne, that actually not only for inviting us here, but they helped to fund some of our original research at NCH to get this entire field moving. But thank you.
Thank you, Teji. Sorry, what do I do to advance? Is the green button? Okay. I'm Dan Levy. As I mentioned, I lead the gene therapy program for Duchenne muscular dystrophy at Pfizer. And I'm going to be talking about our gene therapy, which is an AAV replacement gene therapy called fortadistrogene movaparvovec, as well as talking about some of the contributions that you folks in this room have made. I got a lot to cover. I'm going to dive right in. This is an overview of our clinical development program, all with fortadistrogene. And I'm going to focus on 3 boxes that are there on the left side. The first is our phase 1b study that was started several years ago in boys aged 4 through 12 years old. There were 19 ambulatory boys in that trial. The second we began about 2 years ago. It's a phase 3 double-blind placebo-controlled trial in boys aged 4 through 7. That's called CIFRIO. And then we most recently started a trial called the Daylight trial in boys who are aged 2 and 3. And I don't think we've shared this yet with anyone, but we have completed screening of the CIFRIO trial. So we've identified all of the patients for that trial, and also have identified all the patients for the Daylight trial as well. So thank you to those of you in the audience, you families who may have participated in some of our trials. And also thank you very much to Cure Duchenne, because like the Sarepta program, our initial work, which was led by Jude Samolsky, was funded in part by Cure Duchenne. So we're really very appreciative. This is a brief summary of what I'm going to share with you from our trial. I'll be talking about the safety profile. I will share with you the amount of mini-dystrophin that's expressed, that's seen in those muscle cells. And I'll also talk to you a little bit about what we believe is a functional improvement relative to natural history that we've seen in this open label trial. Okay. So this is a description of the open label phase 1 trial. Again, there were 19 boys in this study, and if you look to the left side, you'll see the requirements or the inclusion-exclusion criteria for the trial. On the right side, you'll see the endpoints that we were looking at over the first several months and up through 12 months of the trial. And in the schematic below, if you look to the left side, you'll see that we dosed the first 3 participants with low dose treatment, and the remaining 16 got high dose, which is 2 × 10 to the 14th particles per kilogram of fortidistogene. And in this trial, we collected data on function as well as biopsy at baseline, biopsies also at 2 and 12 months, and functional data along the way. So this is my first slide here on the biomarker of miniaturized dystrophin, or mini-dystrophin. On the left side, we have the concentration of mini-dystrophin that we measured from the muscle biopsies. In comparison to the normal amount of dystrophin that's expressed in a young man's muscle, we saw essentially 0 of the dystrophin or mini-dystrophin at baseline, and by 12 months we saw approximately 30% of the normal amount of dystrophin that's in those cells, which is, we're very pleased by that result. That's quite a bit of the protein. And then on the right side, you will see the percentage of fibers that under the microscope were glowing, were lighting up, showing that they had mini-dystrophin inside them. And you see there that by 12 months on average, we had more than 50% of the muscle fibers had the mini-dystrophin inside them. So I'm going to switch by talking about function, but I've got a busy slide to go through first. This had no placebo in the trial, so it's an open label trial. Everyone got gene therapy. So if we want to compare how the boys in our trial did to, say, natural history, we need to come up with some sort of comparison group. And what we did was we took 2 placebo-controlled trials. There were 156 in Duchenne. There were 156 participants in those trials. And when we matched up the criteria that we had, age and certain functional elements, or functions that these children could perform, we came up with 66 who met those criteria and served as our comparison group. 60 of them had North Star scores collected. And what you can see on the slide is essentially that their baseline features were pretty well matched as far as their functions, as far as their age. So we felt that this is a good group to use as a comparator. And here is my first function slide. So this is a slide looking at the North Star Ambulatory Assessment. The North Star score is made up of 17 different skills, and you can score a 0, a 1, or a 2. A 0 means you couldn't do it, a 2 means you did it fine, and 1 means so-so. So the maximum score is 34. And what we're looking at on the slide is actually the change over 12 months. So we're not showing the score itself, but how it changed from baseline to 12 months. And what you'll see there, the blue dots represent the low dose participants, the green dots, the high dose participants. And in those gray boxes, that's essentially the data showing the results that were collected for the external cohort group. And the black line, the thick black line in the middle of the box is the average score, the average change from baseline to 12 months in the external cohort. So what we see on average is a 1-point increase in the placebo-treated patients, and in the fortidistogene treated patients, and in the external comparator group, an average of 4-point decline. So that gives us, we believe that there's a difference over 12 months of 5 points on the North Star score compared to natural history. Looking at some of the individual skills here, at baseline, about 1/3 of the boys in our trial had at least one of the tests where they scored a 0. And it was a similar percentage for the external cohort. Over the course of 12 months, 2/3 of those boys got a score of 1, at least 1, on that test that they started off with a 0, as opposed to 1/3 of the external cohort. And if we look at either at maintaining your skills, on average, 16 out of the 17 skills were maintained by the gene therapy treated boys. And in the external cohort, about 11 out of the 17 skills were maintained. So overall, we feel that there is some nice evidence that compared to natural history or compared to boys in a placebo-controlled trial, that there is benefit here on their function. I'm going to switch over to safety. I'm running a little low on time, so I'll speed up. On the right side, there's a table, shows the adverse events, which is another word for side effects, for the low-dose patients and the high-dose patients. And on the left side are the serious adverse events, or SAEs. And SAEs are ones, for the most part, where patients need to be hospitalized because of the adverse event. We had, out of the 19 participants, we had one boy who was admitted for nausea, vomiting, and dehydration, received IV fluids. And there were 2 other participants with serious adverse events for kidney injury and low platelets, which we believe are part of this process called thrombotic microangiopathy that Dr. Bonneman mentioned. And that's a very serious immune reaction against the AAV. Thankfully, all of the participants in our trials have recovered completely from that within 2 weeks. But we have worked to either reduce the likelihood of this kind of thing happening or the severity of this kind of thing happening. And I do also need to mention, I think most of you in this room are aware, but we did treat some non-ambulatory participants, and there was a tragic event in a 16-year-old young man with Duchenne who had advanced cardiac disease. And after receiving the gene therapy, he developed severe heart failure and died of shock. So this was really quite tragic. And we've obviously have worked to try and prevent any kind of reaction like that. And I'll discuss that in just a moment. This is just a summary of the different kinds of adverse events or serious adverse events that have been seen across gene therapy trials for DMD. And what's really notable is, for the most part, all of these are different kinds of immunologic reactions against these viral capsids that are being introduced into the body of the DMD patients. So with respect to trying to prevent these kind of reactions, one thing that we have done is increase the amount of steroids that we give the DMD participants in the first several weeks after receiving the gene therapy. We believe that this has reduced the chance of several of these kind of side effects and certainly the severity. With respect to this concern about cardiogenic shock, we're now requiring that all DMD patients who are being considered for our trials are evaluated by a pediatric cardiologist to ensure that they don't have advanced heart disease. And unfortunately, for those who do, at this point, we are not providing gene therapy for them. In the interest of time, I'll just say that there are other ways, again, as far as increasing immunosuppression and monitoring, and in some cases, exclusions around specific genetic mutations that we've employed. So this is a summary. I talked about the safety. I've shown you that there's good amounts of mini-dystrophin being expressed, and finally that we have evidence of improved function as compared to a natural history cohort. So moving on, I'm going to talk about the role that you folks have played here. We have, from the very beginning, we've had patient ad boards, patient advocacy group ad boards, and we've worked with the different advocacy groups to ensure that we're hearing about the needs of the DMD patient in the setting of a clinical trial, trying to make sure that people are aware of our clinical trials so that not only people who are able to come to meetings like this, but people all across the country are aware of these opportunities, as well as making sure that you folks have been informed as safety events have emerged. And specifically, again, several ad boards from caregivers. We meet regularly with a group of patient advocacy organizations. We have a patient on our steering committee, a patient on our safety data monitoring committee, and we've implemented certain changes to our protocols based on all of these feedback. So very grateful to all of you folks in this room here, certainly grateful to Cure Duchenne, and again, all of the providers and patients as well. So thank you very much.
All right, I hope my mic is working. Thank you so much to Cure Duchenne for having Regenxx here today. And I go to my slide here. My name is Jahan Nazdaskir. I go by Naz for short. I am a pediatric neuromuscular specialist by training, who trained under the humble and brilliant Carson Bonneman sitting over here. And I'm currently working as the clinical development lead at Regenxx Bio. So RGX-202 is our investigational gene therapy treatment for Duchenne muscular dystrophy. As you've seen in the prior slides, we also have forward-looking statements that I won't go through. So I'll tell you a little bit about our company because we are relatively kind of the newer kids on the block here. We are obviously seeking to improve the lives of kids with Duchenne through the curative potential of gene therapy. We are a leader in AAV gene therapy with exclusive rights to the NAV technology platform. We have end-to-end capabilities. We do research, clinical development, and commercial-ready manufacturing. We have a broad internal pipeline with expertise across multiple therapeutic areas, including neuromuscular disorders. We have proprietary AAV manufacturing, NavXS platform, and internal manufacturing facilities, as we mentioned. It's on the 4th floor of our building in Rockville, if anybody's interested in coming to visit. We're very excited about that. So RGX-202 is our investigational microdystrophin gene therapy. We've obviously partnered with the Duchenne community to help guide development. We are so grateful to advocacy groups such as Cure Duchenne for their help in this, and all of you who have contributed your feedback. Our Affinity Duchenne study, which is the title of our phase 1/2 study, is actually now active and recruiting patients. So I'll tell you a little bit about our product. So our microdystrophin is a little different from the other ones that you've seen. We have designed it to retain key elements of full-length dystrophin and include coding regions for the C-terminal domain. So you can see what full-length dystrophin looks like over here to the left. And our microdystrophin, also known as RGX-202, is modified similarly to the other companies, but what we've done is we've included some other elements which include the C-terminal domain. So we're hoping that it kind of anchors better to the sarcolemma or the muscle membrane. We have preclinical data in the DMD animal model, which is known as the MDX mouse, that helped support our decision to move forward with human trials. So we have shown robust microdystrophin expression in skeletal and cardiac muscle. So we look at the amount of microdystrophin in the gastrocnemius or calf muscles, the diaphragm, and the heart. And with increasing doses of RGX-202, we see greater levels of microdystrophin expression. We see a significant increase in muscle function and strength with the in vitro force data, which is strength testing of the individual muscles in the mice. Again, with the increasing doses of RGX-202. And at the bottom here, you can see that there's an improvement in muscle fibrosis, which is some of the scar tissue that you sometimes see in the muscle that develops in kids with Duchenne muscular dystrophy. So here on the farthest left, you can see a wild-type mouse which has no mutation. It's not an MDX mouse. And a mouse that has had no dose of RGX-202. And with escalating doses from dose 1 through dose 4, you could see that the percent of fibrosis, or those blue areas in the muscle tissue, kind of diminishes. So we found all of this to be incredibly hopeful for us. So now we'll give you through the timeline of where we're at. So check marks are what's been completed. So we have designed RGX-202, and I'll give you a bit more detail on the construct itself. This is an AAV8 vector. We use an SPC512 muscle-specific promoter, and we have an optimized microdystrophin transgene that includes the coding region for the CT domain. Check. So we move on to the next one. We have preclinical IND enabling activities, so we had to present our mouse data to show that we can transition into treating humans in a trial. And we did that. So we have a manufacturing process development. We've gotten community input. We have a clinical study design. Check. And now we have activated our phase 1/2 study. You can find us on clinicaltrials.gov. The link is on our final page that you can see. And we are active in recruiting patients. So our RGX-202, as I mentioned before, is AAV8 vector, so actually different from our prior speakers. We use AAV8 to deliver the transgene to the skeletal and the heart muscles in the body. We use SPC512 to drive the expression of the protein in the muscles. And our microdystrophin transgene is also kind of included in that package, as I mentioned, again, including the CT domain. So I'll tell you a little bit about our trial. So this is an open-label, multi-center clinical trial to evaluate the safety, tolerability, and efficacy of a one-time dose of RGX-202 in boys with Duchenne. So the key eligibility criteria are boys aged 4 to 11. That's up to 11 and 11 months. We're not going past 12. You have to have a genetic confirmation of Duchenne muscular dystrophy, and we have unfortunately had to exclude some of the exon mutations, as some of our colleagues have done as well, just from a safety perspective, just to start off with, but hopefully that will change in the future. So we're excluding anyone who has exon mutations in 1 through 17, but we're including everybody with 18 and above, deletion, duplication. You should be able to perform a 100-meter walk test without any assistive devices, and that will be assessed at screening to see if you could be included in the study. You should have no antibodies to the vector that we're going to be using, which is AAV8, and we cannot include anybody who's used prior gene therapy as well. Our outcome measures are going to include safety assessments, strength and function as measured by the North Star Ambulatory Assessment, microdystrophin protein in the muscle. When we check on muscle biopsy, we can see that the microdystrophin protein that's delivered by RGX-202 is lining the sarcolemma and actually being delivered to the correct place. And we're going to check for skeletal and heart muscle changes as measured by MRI studies. So we have a screening period that comes in right before day 1 where we give RGX-202. What also happens during the screening period is we do start an immunosuppression regimen, which includes some medications to kind of help prevent some of the immune responses that we can see that can sometimes be adverse events in clinical trials for gene therapy. And then what we're going to do is do a follow-up muscle biopsy at around 3 months and continue to check muscle strength, function, and MRI studies at selected visits over the course of 12 months. The primary endpoint of our study is, because it's a phase 1/2 study, is safety and tolerability. And that's our utmost importance for us to make sure everything is safe, what we're doing. We have a comprehensive immunosuppression regimen as part of the trial. We have a dose selection period, so we're starting off with 2 different, one cohort called cohort 1. Hopefully this is easy to follow up here. So cohort 1 is going to have increasing weights of participants that are dosed at the lower dose. Lower dose doesn't mean less something that you're kind of putting your child into to receive something that's not good for them. This is the lowest effective dose. And obviously we try our best to see how far we can kind of push that without having any safety risk. So we do the lowest effective dose for the first 3 patients. And then those first 3 patients may have the option, after that, there is an option of going into the expanded cohort where we can enlist about 6 more participants where they can get that lower dose. But at the same time, if we pass through our safety committee meetings and make sure that it's safe at the lower dose, we will then escalate to dosing patients with the higher dose. So that's 2e to the 14th genome copies per kilogram. And then once we dose our 3 patients in that second cohort, we can actually do an expanded phase, and we have the capacity to dose 18 patients in this trial. So as I mentioned, we're going to be doing intensive safety monitoring. There's going to be internal monitoring between the first 2 patients, and we have an external committee after the 3rd patient, and we're trying our best to stay as safe as possible with this process. Just a side note as well, beyond our gene therapy study, we have actually opened up another study called Affinity Beyond. This is an anti-AAV8 antibody assessment in individuals with Duchenne muscular dystrophy. So this is a non-interventional study that we've implemented to try to understand the prevalence of AAV8 antibodies in boys with Duchenne and understand who may be eligible for Affinity Duchenne or other clinical trials if you're interested. So it just consists of a phone or video interview and some blood work. We have a central primary investigator. This can all be done at your home. We have clinical nurses that can come out and be dispatched across the country. And key eligibility criteria include boys who are 0 to 11. You have to have a diagnosis of Duchenne muscular dystrophy, and you should not have received any prior gene therapy just because it could definitely skew the result. So summary. So RGX-202 is our investigational microdystrophin gene therapy. We have our phase 1/2 Affinity Duchenne study that is active and recruiting. Thankfully, in partnership with a couple sites that are now open, which is Arkansas Children's Hospital in Little Rock, Arkansas, and Lurie Children's Hospital in Chicago. And just know that we at Regenix are partnering with you, the Duchenne community, to help guide us with the development, and we have an open door policy. Please feel free to reach out to us at any time. We're happy to hear from you. And you can learn more about us at our website, www.regenixbio.com. Here's an email, and you can find us on clinicaltrials.gov for our studies. Thank you.
Perfect. All right, thank you everyone, and thank you to Cure Duchenne for giving us the opportunity to talk today. My name is Patrick Gonzalez. I'm the Senior Director of Clinical Science at Solid Biosciences. And just as my background, I am a muscle biologist and physiologist by training. So for anyone who I haven't met yet, always encourage you to please come up and ask me questions. We've got a short talk today, but always happy to talk more about any of these details, both about the company and the disease. First, I wanted to start here by giving a bit of an overview, an introduction or reintroduction to Solid Biosciences and who we are. For those of you who may know us so far and now who may not. So first, I'll say we are a public company, and so we also have forward-looking statements here. But Solid Biosciences as a whole is and has been initially focused on Duchenne muscular dystrophy and really advancing meaningful treatments since our inception. Throughout the years, we have developed our initial gene transfer therapy, which is SGT-001, which we'll go into some of the details on today. And now more recently, we've evolved in kind of understanding what are the approaches that we can use, the platforms that we can take advantage of for addressing the disease, move from being looking at all opportunities to focusing on microdystrophin, gene therapy in specific. And also in the end of last year, we acquired an additional company called Avanti Bio. And so Avanti Bio was focused very much in parallel with what we were on rare neuromuscular diseases. And so now we'll kind of talk a little bit about it more, but we have an expanded pipeline focusing on Duchenne as kind of our primary and lead candidate still, as well as wanting to move into other neuromuscular diseases and rare cardiac indications as well. So this slide is just kind of looking at what our pipeline is. And I mentioned SGT-001 as our lead program that had gone through clinical development previously. And now today what we'll talk a little bit more about is also SGT-003, which is our current lead program. And so we're kind of in a combined effort with what we've just talked about from the other sponsors here of having clinical data in our previous program as well as updated program now, SGT-003, which is going through IND-enabling studies to now move into the clinic shortly. And so first I'll talk about SGT-001 and what was our Ignite DMD clinical trial. And so Karsten and the others have done a great job so far in really giving an introduction to what is gene therapy, what is a microdystrophin gene therapy, and what are the pieces that you need. So I'll kind of go directly into the details of our approach here today. And so our Ignite DMD study was a phase 1/2 clinical trial. And so it's still an ongoing study, but this was designed to assess the safety and efficacy of SGT-001. SGT-001, taking a look at what are all the pieces of a gene therapy, was an AAV9-based gene therapy approach using a muscle-specific promoter called CK8 to deliver a microdystrophin transgene. And so we'll go into a little bit more details on what our transgene is later. But overall, for this IGNITE study, this was evaluating that specific construct, which we consider kind of our first-generation gene therapy approach here. As you can see from the listing, we dosed 9 patients in this study. It was a dose ascending study, and so we did look at both a lower dose of 5E13 as well as a higher dose of 2E14, which our study was escalated to after looking at microdystrophin levels. We did initiate in non-ambulatory patients and then moved into ambulatory patients as well. And so that's kind of important for kind of what have our learnings been along the way, because overall, as everyone knows here, we want to be able to treat as many patients as possible and expand those cohorts. Since this was a phase 1/2 study, we focused on safety as a paramount importance, and then also looking at what are we seeing in microdystrophin expression, what are we seeing in functional changes in the patients as well over time. So the enrollment in this study has concluded, and we're continuing our 5-year monitoring for post-treatment as is required by regulators, and look to kind of update at the end of that study as well. And so what was highlighted today, and really what I want to kind of focus on for how we're using the data that we've collected and been able to learn throughout this study, is what's normal and kind of expected versus what else do you need to still be able to focus on and learn more from in terms of safety along the way. And so I think what's been very common amongst high-dose gene therapy trials has been really looking at this corner here of the most common drug-related clinical adverse reactions. And so if you take a look here, you see nausea, vomiting, and fever. And so these are kind of things within those first few days, as was outlined before, that really is kind of what's seen normally through most of these different trials when you're at these high-dose levels. Looking at our specific trial, we did have SAEs as well, so AEs, adverse events, and then also looking at SAEs, serious adverse events, which we saw were related to complement activation. And so by thinking about how are you getting that complement activation from our research, and both in the clinical side as well as non-clinical looking at lab results, we see that it's really related to the amount of capsids that you have and viral particles, as well as having that together with the antibodies coming up over time. So it's something that we're looking at further and really drove where our innovation has gone to from our first generation to next generation product. And so what we can say also is that we did see these SAEs that did occur early after infusion in the first few weeks, but none did occur after 90 days post-infusion, and all these patients did recover fully, and all the SAEs have resolved, and so they're still going through their post-treatment monitoring now, from over 1 year post-treatment up to 5 years in the study. So this study did initiate 5 years ago, so we're very lucky to have had patients on the study for that entire period, so always very thankful to the entire community and patients for that. And so looking at what the other speakers have also kind of focused on is obviously we want to see what's the immediate effect of your treatment when you're actually administering your gene therapy, and it's your expression of your protein. And so we do 3-month and 12-month biopsies, so wanting to look at what is that short-term expression that you're seeing, and is it also lasting over time. And so we can see by using Western blot here, and then in the picture here, which is immunofluorescence imaging for microdystrophin, you have positive microdystrophin staining at the muscle membrane, which is showing you that the microdystrophin is functional. It's also distributed throughout the muscle. And then also in terms of just the levels that we're seeing of dystrophin, we see that these levels are sustained over time from the 3-month to 12-month time points. We also had a unique kind of ability in this study because of COVID and the inability to get all the patients in for biopsies at the same time. We were able to get a bit longer term time points as well, so looking at 18 months and 24-month time points. And what we were able to see is that when you look at those individual patients, they really had kind of sustained levels of expression over that time. So initial indication, of course, very small n numbers, but that we may have something that could have a durable result. And so just quickly also looking at the function from this study, what we saw was that in that 1-year time point, which is really a primary analysis time point of the study, we evaluated motor function, pulmonary function, and then also included the PODC, which is the pediatric outcome domain, which is a patient-reported outcome measure. And so we saw that we had positive changes from that baseline to 1-year time point in each of these evaluations that really went hand in hand with one another. We also had a control group in this study, which had patients who were on an initial delayed treatment. So we were able to see that those patients did decline over that time point. However, we saw positive changes over 1 year for the treated patients. And so kind of what does that take us to? And for us as Solid, we want to be able to understand what we learned from that study from the safety side, what we learned from our construct, and potentially seeing some initial signs of efficacy from our microdystrophin expression, but then using what we've done in the background and really looking to evolve and really wanting to look at, can we do something more in the lab? Can we do something more with our approach to be able to optimize what our strategy is and be able to then move into a next product? And that's really where SGT-003 came in. And so this was really all based on our focus on a gene therapy and the AAV-based platform, so adeno-associated viruses. We talked about how there's different serotypes for each of these capsids. They have more or less efficient targeting of different tissues. And so we initially used the AAV9 capsid, and we took a step further and wanted to be able to see, can we do something more to optimize that capsid and see, can we actually get better muscle targeting by changing pieces? And so in our lab, we developed a novel capsid discovery platform, and also were able to now from that platform choose lead candidates, which we moved from in vitro testing to initially look at, are you seeing improvements in the amount of expression that you have in your muscle and the amount of actual viral particles that get into muscle? Then move that into non-clinical testing as well, so using the MDX mouse model, which is dystrophin negative. And what we can see when we're comparing our novel capsid, and here, which is specifically going to be SLB101, which is a capsid that we've named as our lead and is now in our SGT-003 program, we can see that comparing that to AAV9, we really do see a multiple-fold improvement in the level of expression that we can achieve by administering an equivalent dose level. And so here, looking at kind of that immunofluorescence in the center panels, and then what's using a reporter on these right panels. So you can see just when you have this, this is a luciferase reporter, and so basically the higher amount of expression is coming up as brighter red. And so you can see by using this novel capsid at the same dose level, you have a higher amount of expression. And for us, that was a meaningful and a big reason to really advance this forward. We took it a step further, and we also looked at how does this translate into larger species? Is this something that we're expecting to see maintains that consistency of improvement as you get bigger and bigger? So we did a non-human primate study as well initially during our research efforts to show, are we seeing that similar fold improvement over AAV9? And the answer is that we did, and we saw that there were approximately 2 to 3 fold improvements, at least in our biodistribution and expression levels that were to that extent or better. Importantly, we also looked at the non-specific effects of this capsid. And so while we're improving the muscle targeting side, we also wanted to see how that impacts our non-muscle side. And so what was mentioned before is that these kind of capsids will go, you're obviously administering systemically, so they're going to go everywhere in the body. If you can limit the amount of capsids that are going to the tissues that don't need them, that can be a positive. And so what we saw is that we see about half of the amount of capsids going to the liver especially, compared to AAV9, which is a big pro for us. And so all of that put together this program, which is SGT-003 and is currently in IND-enabling studies to be able to hopefully move into the clinic as soon as possible. It utilizes this optimized transgene that we use for our SGT-001 program. Unique to our transgene, and we kind of went through it before, and Carson did a great job of outlining the different ones. Unique to ours is the inclusion of this nNOS binding domain, so R16, R17, which we've seen is able to restore nNOS, which is neuronal nitric oxide synthase, to the membrane, something that could be important for preventing activity-induced ischemia, for potential additional protection of the muscle. Using this capsid, where we've seen multiple fold improvements, as well as an updated manufacturing process where we also see improvements over our past process that we used, we see that there could be additive benefits of kind of our overall new strategy for SGT-003, and that's why our focus is entirely on this program at this time. And so these are kind of the buckets of things that we're doing right now, and there could be very, very long lists under each one of them, but we'll keep it high level. Right now, we're in those IND-enabling studies going through the efficacy studies, safety testing, most importantly, to be able to establish our full package to move for FDA to put in front of them for the IND, submitting that IND at the second half of this year, and then targeting the initiation of our clinical trial later in 2023. So I'm not going through the details of what that study is looking like right now. We're obviously looking to go in with kind of a similar similar approach of going smaller and learn more and expand from there. But the details of that study, as we talk to regulators and kind of refine these things with these great consortium that we've established to understand safety, mutations, and different approaches that you might need, are going to kind of be what builds that final strategy. So with that, I just want to thank you for all your attention. Thank you to everyone who's participated in the study, as well as all the family and community out there. And as I said, always happy to take additional questions if you see me in a corner later. Thanks.
So hi, so I'm actually going to switch things up a little bit now and move into a slightly different approach that we're considering for Duchenne muscular dystrophy, and almost kind of a next generation approach, so to speak, and that would be gene editing. Before I go into some of the details around what we're doing on the gene editing side of things, I would like to introduce Vertex. So as you could see from my introduction earlier, I am an employee of Vertex Pharmaceuticals, as you can see from my disclosures and disclaimers side. So who are Vertex? So Vertex is a global biotechnology company that really invests in scientific innovation to create transformative medicines for people with serious diseases. We take great pride at Vertex that patients are the heart of everything that we do in the company, and we really aim to strike at the core of serious diseases to change people's lives. And with that, we're also not afraid to take on the impossible as we think about the diseases that we work on. So we tackle tough diseases. We do that by really spending time to understand the biology of the disease, have a really deep understanding of the disease itself, how the disease manifests, what's the biology behind that, so that we can really develop the right treatment approach that not only has the potential to increase our probability of success, that treatment will be successful, but that treatment will be transformative for that disease. So not incremental, not potentially just treating the symptoms of the disease, but really treating the root cause of the disease. So what does our strategy look like? As mentioned earlier, Vertex are known as pioneers in the cystic fibrosis space. We discovered, developed, and produced the first medicines that really address the root cause or the underlying cause of cystic fibrosis. We are now taking the learnings in the cystic fibrosis space, and we are building upon that as we move into additional and other diseases. In order to do that, though, we're also investing in other treatment modalities or different approaches to treat diseases, and that really includes complementing Vertex's experience in the small molecule side of things, so oral medicines, pills as you would, into cell and genetic therapies. And over the last couple of years, we've really built up our Vertex cell and genetic therapy side of things, or VCGT, as we call it, pulling together a portfolio of different technologies, so addressing the potential to use genetic medicine as well as cell therapies. The teams that we're building within Vertex and making sure that we have the right people involved with the expertise to develop the right approach and then to very quickly move that approach towards the clinic, and as well as manufacturing, so that we are ready to move this forward quickly. And we really believe that a number of diseases in our pipeline, including Duchenne muscular dystrophy, really has the potential to be transformed by using cell and genetic therapies. And as mentioned at Vertex, we really take a disease-first approach to everything that we do. When we decide to work on a disease, we really investigate that disease from every angle. And as mentioned, by doing that and understanding the biology of the disease, we really design the right treatment approach for that disease and then build the toolbox to potentially then build the treatments and then develop them further. So just very quickly to show a little bit about our journey in cystic fibrosis, because again, this is how we think at Vertex, and in some respects, this is how we're thinking about what we do in Duchenne muscular dystrophy. As mentioned in the panel earlier also, Vertex has invested in cystic fibrosis for more than 20 years. We now have 4 approved medicines that treat up to 90% of patients with cystic fibrosis. As mentioned earlier also, that wasn't initially the case. Initially, we started with a small subset of patients, but over time built therapies that really targeted other subset of patients so that we could build up, so that we could treat as many patients with cystic fibrosis as possible. But we don't stop there. We realize there's still 10% of patients with cystic fibrosis that still don't have a treatment approach or a treatment modality that might work for them. So we continue to invest in that research. And as such, we actually still have upwards of 20 clinical trials ongoing in the cystic fibrosis space. As we think about cystic fibrosis and then applying that to Duchenne muscular dystrophy, you can see here that it's really all about commitment, expanding access to all patients, and in the cystic fibrosis side of things with the existing medicines or potentially new medicines, continue to out-innovate ourselves. So as we also spoke about earlier, with that innovation side of things, we don't just stop with a medicine that we have. We need to continue to innovate over time and continue to develop better medicines, learning from what we've done and applying those learnings to the next generations of medicines for patients. And in cystic fibrosis, we actually are also investigating genetic approaches. So at Vertex, we have the opportunity to leverage our understanding in the genetic therapy side of things and in the cell therapy side of things to apply across multiple programs. So with that, and with that journey in cystic fibrosis, Vertex has really now built a broad pipeline with multiple diseases that we're working on, with Duchenne muscular dystrophy and myotonic dystrophy, actually, in our muscle disease side of things. So with that introduction, I'm now going to pivot and talk more about gene editing for Duchenne muscular dystrophy. As you heard this morning, from my fellow panelists, there are multiple different approaches to gene therapy. 2 of them are shown in this slide: gene addition or gene replacement, which is the approach that's been taken by multiple sponsors looking at, for example, in the microdystrophin or the mini-dystrophin gene therapy programs. As you heard this morning, gene addition or gene replacement therapy involves adding an extra copy of a healthy gene into a cell to replace the faulty gene. And with that, to be effective, that additional gene really needs to be expressed over the entire lifetime of the patient so that it can continue to supply the healthy version of the gene that is faulty. Gene editing is slightly different, where with gene editing, we have the opportunity to fix the error or fix the faulty gene at the level of the DNA. And we can do that by disrupting the gene, deleting part of the gene, correcting the gene. And as you can imagine, by using gene editing, that really has the potential to result in a permanent change to the gene and to the cells that have been affected by the gene editing. Understanding gene editing a little bit more, gene editing can also be called genome editing. You can use that term interchangeably. And it really refers to a group of tools that scientists have developed that allows us to change DNA. As mentioned on the other slide, those tools can be designed to disrupt, delete, correct genes at very precise locations in the DNA. So in the case for Duchenne muscular dystrophy, that would be at precise locations in the DMD gene. Several approaches to gene editing have been developed Several of them are actually in the clinic being investigated for other diseases. But a recent one that you may have heard of and be familiar with is CRISPR-Cas9. So what is CRISPR-Cas9? So CRISPR-Cas9 is a gene editing tool that allows for precise gene editing, i.e. the CRISPR-Cas9 gene editing machinery can be targeted to a specific gene. There's really two main components of CRISPR-Cas9. The first is the Cas9 protein itself, which is actually the protein that can actually cut the DNA. So sometimes you may have heard that referred to as the molecular scissors of the gene editing machinery. The second very important component to CRISPR-Cas9 gene editing is a guide RNA. And a guide RNA is a unique barcode of sorts that shows exactly where in the DNA or where in a specific gene that Cas9 should make the edits. So in some respects, Cas9 can also be referred to as the GPS of the system. So that's basically what directs the gene editing tool to make the edits at that precise location within the DNA. How are we thinking about using this for Duchenne muscular dystrophy? Now, one potential approach here is to use gene editing to correct the reading frame at the level of the DNA at the DMD gene. So as you know, there's many mutations that cause Duchenne muscular dystrophy that puts the DMD gene out of frame. And what does that mean? The reading frame is basically where the DNA bases are put into what we call codons that are then made to translate into amino acids and then protein. And when you have a mutation that puts the DMD gene out of frame, it means then that that protein can't be produced, and in the case of DMD, it's the dystrophin protein. We can design CRISPR-Cas9 gene editing to make very specific edits in a DMD gene that can put the DMD gene back into frame. So we call this reframing. And when you reframe the dystrophin gene, you then have the potential to then produce protein, and in this case, it would be near full-length dystrophin protein. So this is a schematic of some work and some research that we have ongoing at Vertex in our mouse models of DMD. And this really shows how we can apply gene editing and what some of our results can look like. So obviously, we need to deliver the CRISPR-Cas9 gene editing machinery to the target cells, so to the skeletal muscle, the heart, and the diaphragm. We're doing this at the moment using AAV, so we can actually fit the CRISPR-Cas9 gene editing machinery into the AAV genome for the virus to then deliver that machinery to the cells. As mentioned, we've designed the gene editing to then make precise corrections at the level of the DMD gene, so at the level of the DNA. And you can see here that in our mouse experiments, we can show that that really results in very robust restoration of dystrophin protein expression in the muscles of the DMD mouse models. So obviously, this is a next-gen, an innovative approach. So where this program is at the moment is that we're currently in IND-enabling studies, which means that we're looking very carefully to characterize the efficacy with the gene editing approach, as well as, very importantly, the safety of the approach. And those studies are ongoing at the moment, and it's the results of those studies that will need to be obtained before we can move this into clinical trials. So with this, I'd like to finish by inviting you to, if you have any questions or want to learn more about our DMD program, please visit the Vertex website or follow us on social media. And thanks very much.
Thank you all. Thank you to all our panelists. We're actually going to open it up for just a few questions. If we have some from the audience, I know Jennifer and Doug are running around with microphones, and I can't see great. So if Jen wants to start. We only have about 10 minutes here for our questions. I know there might be a lot, but our speakers today will be here for the rest of the day and throughout lunch and everything, and might be able to answer those offline as well. Jen, do we want to get started with a question? Oh, you're right there. Thank you.
Hi, Jen. First of all, thank you all for all you're doing for our boys. We really appreciate that. Everyone, round of applause. Yeah.
Yes, thank you.
And I know I'm not alone. My son has a lower deletion, like it's actually 1 to 64. It's very unique. But for those who have deletions below 18, any words of wisdom or anything, any hope for that to boys to be able to be treated within a reasonable time frame? As my boy's 13 and a half, and I know time's of the essence for all of us. So any ideas on that?
So I make a high-level comment and then pass it on. So your deletions below 18 is your questions, yeah. So there's in our group that are represented here on this stage, there are efforts going on to understand the fine anatomy of those deletions. So it's probably not that every time you have a deletion that overlaps with the transgene, you're going to have the same immunological risks. There are probably parts of the dystrophin transgene that are particularly prone to doing this, that are particularly immunogenic, if you will, and others that are less. And there are probably some, as I mentioned this morning, there are probably some immunological constellations in the patients themselves that make them higher risk of having that and lower risk. And the idea going forward is to understand the details of these risk equations and then have a proactive management, proactive treatment for each of these scenarios at hand, where you can immunosuppress the patient proactively to make sure that you cover this patient's individual risk appropriately. So that will be going forward with the same transgene, the same methodology. You can also think about making transgenes that are less immunogenic by just kind of re-engineering them and making them less immunogenic, although that's a much longer developmental process, of course, because you have a completely new product. But I do see, and I'm curious what others have to say, I do see a way forward with the current constructs, with the current approach, by treating the patients according to the immunological risk that we assign to them proactively with an immune modulation protocol so that the immune response is suppressed when the transgene comes up. And in the long run, that tolerance is happening. So, but what the immune system does, fortunately, it learns. So when there's a foreign antigen there and you suppress the immediate rejection, the immune system will then learn, oh, this is now there, this is part of the body, we're going to tolerate it. It's called tolerance. And so on. Over the long run, you can then hope to get rid of the immunosuppression and have that protein be accepted by the body. That's the way forward.
Sure, I'll add a little bit. I mean, Dr. Bonneman's covered it all. I mean, basically there's three things. Where's the risk region? Who's at risk based on patient factors like HLA typing potentially? And then what can you do to do the risk mitigation? So under his guidance, we've actually all worked together to try and identify some of those regions. So we do expect some more work earlier this fall about maybe better identifying subjects, like patients themselves, what are their risk driving factors. On the Sarepta side, we have done studies in our 103 one where we expanded the 1 to 17. So we've looked at subjects, patients with mutations in that region to really understand the better areas that are highly driving it. The 1 to 17, just to bring it to historical context, that was Dr. Mendel's work in 2010 that had already pointed this out. And Dr. Flanagan did some work in this area as well. And then I think now we're just getting more specific on where those patients are and then characterizing them between low, medium, high risk. And if we identify someone that's at high risk, Dr. Barry Burns and company in Florida, they're thinking of ways to how to actually suppress the immune system so you can tolerize the patient pre doing it. So I do expect that a lot of this will change. I don't have a timetable, but I think this is going to evolve pretty fast.
Yeah, again, coming back to my example of this morning, you would never do an organ transplantation in a patient without immunosuppression, right? This is now common, this is routine. I think we'll have routine protocols in gene therapy as well, according to the risk that we assign.
Thank you. All right, let's move on to the next question. Doug, do we have one, Jen?
I have one here.
Okay. Thank you.
Yeah, thank you so much. And I've got a quick question with regards to some of the information that we've kind of read secondhand on some of the FDA regulators and their concern with the gene therapy. As a father looking at this, if the data, the publicly available data, show that there's an induction of dystrophin as well as a statistically significant improvement in the North Star, what else can we show? I mean, what other data are there to support that it's functioning? Where would we go from here, I guess?
Go ahead, Dan.
I'll take that one. One of the things that the FDA may choose to wait for will be placebo-controlled data. And Sarepta is in the process. Their study is well underway. Our study is now underway. And so there are differences when you compare to a natural history group and to patients actually receiving placebo in a trial. There is a lot of literature there on how things can be potentially influenced, not intentionally influenced, but for example, boys knowing that they receive gene therapy may perform a little better on those tests. And the boys who are entered into these particular clinical trials might not be exactly the same as ones that are being followed by natural history. So while we feel that the data that we have is compelling and really signals the benefit, it will be up to the FDA as to whether or not they choose to offer accelerated approval based on the open label data that's been shared, or if they will say that they need to wait until the placebo-controlled trials are finished and they can use that, I'll just say, gold standard type of data, which they feel would be more rigorous. And I will note that there have been open label trials that have been convincing in the past, but when placebo-controlled trials are completed, it's recognized that perhaps the treatment effect that was seen is not as impressive as appeared in that open label kind of trial. So I've shared with you, perhaps it's the FDA's perspective on rigor for gene therapies, which do have some safety challenges associated with them. And that perhaps is the rationale by which they, well, that's the structure, that's the trade-off of decisions that they will make within the next 6 weeks.
498 hours now.
498 hours and I apologize for speaking about Sarepta there, but.
No, and I guess my point, as all of you up there are professionals who spent your life dedicated to this. Do you feel strongly, thank you, Jen, do you feel strongly that induction of dystrophin as well as improvement in the North Star would be enough to say that this is scientifically working?
I'll let some of my other colleagues comment on that.
Yeah, I mean, I think from a scientific perspective, it's exactly why we've established a microdystrophin. It's putting together a rational design of the domains that are required to be able to confer function. So from that perspective, I think that's kind of why this strategy is being used in the first place. I think for each program, it's going to be about putting together the data set that kind of proves that as well. We did touch on it a bit before, that every microdystrophin is a bit different, and it's different than a full-length dystrophin. So you do have to put together a little bit more to kind of establish that connection, which is understandable. And I think that's especially because we're in a gene therapy where there are safety considerations that you have to acknowledge, and you have to make sure that you are giving the best benefit that you can. That's why that extra level is necessary. But I agree that scientifically, that's why we're doing what we're doing. And then now we have to kind of understand what's the bar of expression and of the full data set to be able to get an approval.
Yeah, the key really is you have to show at some point that the biomarker, the dystrophin expression, is linked to function. And once you've established that, you'll be quicker with using the biomarker. Give you an example. There are certain mutations in dystrophin where you have such a small dystrophin, it's so short that it actually is counterproductive and makes things worse. And so if you have a high expression of that protein, it does not correlate to function at all. It correlates to loss of function. So you have to really make you can't jump to the conclusion that what you put in is helpful unless you show it once, and then you can use it.
I think the other thing that I would add to that as well is that why sometimes it takes so long for some of these therapies to get to the clinic is because we do an extensive period of preclinical testing, and that's where we also have the other opportunity to show function. So a mouse is not a human, obviously, but there's a lot of studies and experiments that can be designed to show what you're expressing or what you're restoring. In our example of restoring dystrophin expression through gene editing, we can show in our mouse models and in other animal models that that function is there and that benefit is there. It can change the progression of the disease, it can change the pathology of the disease, and all of that builds up into that package to support the function when we move into patients.
Thank you.
There's one more. Do you anticipate, if we get accelerated approval for the first gene therapies, do you think there will be a label exclusion for these first 17 exons?
So I can't comment on what the agency will or will not do. I mean, they have the task of determining the appropriate benefit risk, and the final label would be a discussion at the time that they're favorable for an approval. That said, our own position will be that we actually have indicated our proposed label will have a contradiction or contraindication to 9 to 13 until we learn more at the first stage. And then that's only because of the abundance of caution. So again, the agency will have to make that final determination.
All right.
Carrie?
I know
Carrie, do we have time?
Questions. We are out of time for these questions, but I do ask, come ask a Cure Duchenne staff member, and then we have our representatives here for the rest of the day. There's also an exhibitor showcase happy hour where we'll kind of get to mix and mingle at the end of the day. So please keep these questions close. There's plenty of people to ask. We just have a tight agenda today. There's so much to get through. So I do want to say thank you to all of our panelists on the gene therapy and gene editing session. And you can go ahead.